I am Shuhaib P, Assistant Professor in Microbiology, Unit of Medical Laboratory Technology, Yenepoya School of Allied Health Sciences. Today we are going to discuss about Introduction to Bacteriology. At the end of the session, students will be able to explain morphology of bacteria, describe the bacteria undergoes multiplication, define and explain phases of growth curve and explain factors affecting growth of bacteria. Before that, we should know what is a bacteria. Bacteria are the microorganisms. Microorganism means those are the organisms, living organisms which we can't see through our naked eye, which we can see through only microscope. So bacteria are unicellular organism and it is prokaryotes. Prokaryotes means definite nucleus is absent. So we can see microorganisms only through microscope. So example is bacteria. So bacteria is a unicellular prokaryotic organism. So coming to the shape of bacteria, bacteria mainly exhibit in two shapes, that is cocci and bacilli. Cocci means round shape or oval shape cells and bacilli is rod shaped bacteria. And cocci can arrange in groups, that is clusters, pairs, chains, octet, tetrads or etc. So bacilli can arrange in chains or some bacilli are curved comma shaped. Then coming further, cocci and bacilli we can classify it into gram positive cocci, gram negative cocci, gram positive bacilli and gram negative bacilli. It is mainly on the basis of gram staining. So gram staining is a staining techniques mainly we are using to see the bacteria. So bacteria is a colorless, whatever stains we are using, the bacteria will take up all the stains. For example, if we are using methylene blue, then the colorless bacteria can change into blue color. If we are using crystal violet, and the color of bacteria can change into purple color or any other stain, whichever stain we are using, the bacteria will take up the color of that particular stain and it will appear in that color. So gram staining is a differential stain. Here we are using 4 steps are there. In primary stain we are using crystal violet, in secondary stain or modern we are using as gram staining, and in decolorized reagent we are using acetone alcohol, and counter stain we are using safranin or carbonyl fission. So gram positive bacteria appears in purple color and gram negative bacteria appears in pink color. So some bacteria like mycoplasma, rickettsia, chlamydia and mycobacteria, so these bacteria we can't use the gram staining. Majority of bacteria we can use the gram staining. So for example, gram positive bacteria include staphylococcus, streptococcus, then gram negative bacteria include Neisseria species like Neisseria meningitis, Neisseria gonorrhea, and gram positive bacteria include Bacillus, Corynebacterium, and gram negative bacilli includes E. coli, Klebsiella, Salmonella, then Acinetobacter, etc. Then this I already explained. Then gram positive, the top one is gram positive cocci. Gram positive cocci means purple color round shaped bacteria. It can arrange in clusters, chains, tetrads, octet or pairs. For example, Staphylococcus aureus and Staphylococcus species are arranged in clusters and Streptococcus species are arranged in chains. And gram negative cocci, it can arrange in pairs. In case of Neisseria, it is arranged in pairs and bacilli in chains. Example is Bacillus, or cuneiform patterns you can see in Corynebacterium. And gram positive bacilli, the comma shaped bacilli is Vibrio, and E. coli can arrange in simple bacilli form, and spirally coiled bacilli, that is called as spirochetes, example Treponema and Leptospira. Then this is the bacterial cell. Comparing to the human cells, bacterial cells are unicellular and it is prokaryotic in nature. Here we can't see any nucleus. In case for comparing the human cells, we can see mitochondria, endoplasmic reticulum, nucleus, everything. But here all those things are absent. Here the outermost layer is called as cell wall, and under cell walls you can see a plasma membrane and cytoplasmic matrix. Inside cytoplasmic matrix you can see nucleoid, ribosome, inclusion bodies and mesosome. And ribosome, mainly we are using main function of ribosome is protein synthesis, and mesosomes for cell respiration, and nucleoid, it is the region where the genetic material is present. Then outermost layer is cell wall giving almost the protection to the cells. And in some cell wall appendages you can see like flagella, then fimbriae and capsule. And this is not present in all bacteria. All bacteria contain cell wall, plasma membrane, inclusion bodies, ribosome, mesosome and nucleoid. While some bacteria you can see flagella. Those bacteria are motile. Some bacteria we can't see flagella, those bacteria are non-motile. For example, E. coli and Vibrio cholera, these having flagella, that means those bacteria can move. Then capsule is the outermost layer of cell wall. It is giving extra protection to cell. So this is the overall idea about the bacterial cell. Then coming to the bacterial growth requirements, water, 80% of total bacteria is composed of water, and minimum nutritional requirements for the growth of bacteria include carbon dioxide, nitrogen, hydrogen, oxygen and some inorganic substances. And some fastidious bacteria will not grow in ordinary culture media, they need extra nutritional requirements, then only those bacteria can survive. Then coming to the cell division of bacteria, mainly bacteria is divided by the process, bacteria divided by a simple process. That process is called as binary fission. It can be nuclear division or cytoplasmic division. We know that bacterial cell containing cell wall, plasma membrane and cell wall matrix. The first step of binary fission is the genetic material. The chromosome is present inside the cell wall matrix and it begins replicate and it moves rapidly towards the each end of the cell wall. In this case, the cell wall or cell will elongate and replication will continue, and cell wall also elongate, and at that time the mesosome will invaginate inside and a septa will form. The plasma membrane grows inwards and new cell wall is deposited. In that time a septa will divide a bacteria into two equal daughter cells. This process is called as binary fission. First, genetic material will replicate and it will move towards the bacterial cell, and cell wall will elongate, and plasma membrane and mesosome will membrane will grow inwards and a septa will form, and after the formation of septa, one mother cell is divided into two equal daughter cells and each having same genetic material. This process is called as binary fission. So coming to binary fission, next we should know what is generation time. Generation time is a time required for a bacteria to give rise to two daughter cells under optimum condition is called as generation time. So it is a time, or else otherwise we can call it as it is a time period between two successive cell division. If one bacteria will divide to two bacterial cell and two bacterial cells is divided to 4 and 4 will divide to 8. So it is a time period between two successive cell division, and this generation time period is different for different bacteria. Most of the pathogenic bacteria having generation time like 20 minutes. For example, E. coli. In case of Mycobacterium tuberculosis, it is 20 hours, and Mycobacterium leprae is 20 days. That means for each bacteria generation time is different. Next is bacterial count. You should know what is total count and what is viable count of a bacteria. Total count means it indicates the total number of bacteria. It includes live and dead cell in the specimen, and it is done by the counting bacteria under microscope using counter chamber. And viable bacteria means we are measuring the viable count of bacterial cell present in the specimen, and it is obtained by pore plate method. Next important topic is bacterial growth curve. And bacterial growth curve means when a bacterium is inoculated into suitable liquid culture media and incubated, and the bacteria will grow in that culture media. Then this bacterial count of such culture is determined at different intervals and plotted in relationship time. We will get a curve that is called as bacterial growth curve. So if you are inoculated a bacteria into a suitable liquid culture media under optimum condition, then the bacteria will grow. The bacteria will take up all the nutrients present in the culture media, and the bacteria can grow, and the bacteria will multiply by binary fission. Then bacterial growth curve contain 4 important stages, that is lag phase, log phase, stationary phase and phase of decline. The first phase is lag phase. So when a bacteria is inoculated into a suitable culture media, the culture media containing nutritive substances which help the growth of bacteria. So the bacteria will take up all the nutrients present in that culture media, and then the bacteria will the size of bacteria will increase due to the accumulation of enzymes and metabolites. Then bacteria will reach its maximum size at the end of lag phase. In lag phase, there is no multiplication of bacteria. Bacteria will take up all the nutrients present in that particular culture media, and it will reach its maximum size due to accumulation of enzymes and metabolites. So we will get a flat line in case of total count and the viable count, because there is no increasing number of bacteria, but there is an increasing size of bacteria. Then next phase is log phase. After the end of lag phase, bacteria reach its maximum size, and in case of log phase, the big bacteria will multiply into 2 daughter cells. In this log phase, the bacteria in smaller size, and it is biochemically active, and it is uniformly stained. So here we are seeing in the viable count and total count, there is an exponential growth of bacteria. So there is there is an exponential growth, and you will get a straight line instead of a flat line. That means there is an increasing number of bacteria according to time. Then next is stationary phase. After some times there is a depletion of nutrients present in the culture media. Bacteria will take up the nutrients in that particular culture media, and in case of stationary phase, there is an equilibrium between a newly formed bacteria and dead cell. For example, if 5 bacteria is newly formed, at the same time 5 bacteria will die. That means the viable count of bacteria will be 0. 5 minus 5 is equal to 0. In case of viable count of bacteria, we will get a flat line. But in case of total count of bacteria we will get a straight line, because total count means it is a number of viable count living cells as well as dead cells. So in that cases, viable count of bacteria will increase, and in case of total count of bacteria will increase, and viable count of bacteria will be flat, because it is and there is an equilibrium between newly formed bacteria and dead cell of bacteria. Bacteria will start dying due to the accumulation of there is a depletion of nutrients in that culture media. Then in this phase, bacteria become gram variable, more storage granule is formed, and sporulation occurs in this phase, and bacteria produce exotoxin, antibiotics and bacterioxin. Then last phase is decline phase, and bacteria stops dividing completely because there will be a depletion of nutrients and accumulation of toxic products. So that means there will be all the bacteria present in that particular culture media will die, and so there is you will get a phase of decline, because viable count we are counting only the living bacterial cells, and total count will be flat because there is no more bacteria is forming, but total count we will count dead cells also. In this inversions forms are seen. So here we can see a table. Then in case of lag phase there is no multiplication of bacteria, but in case of log phase and stationary phase there is bacterial multiplication, and in decline phase also all the bacteria present in that culture media will die, so there is no multiplication or division of bacteria in case of decline phase. Then bacterial death, in case of lag phase and log phase there is no death of bacteria. Lag phase, bacteria reach its maximum size. Then in log phase, the bacteria will divide into two daughter cells. There is no death of bacteria, but in case of stationary phase, there is death of bacteria. In stationary phase means it is an equilibrium between newly formed cells and dead cells. So there is a death of bacteria, but in case of decline phase, there is all the bacteria present in that culture media will die due to the depletion of nutrients and accumulation of toxic substances. And total count, in case of lag phase it is flat, log phase also rise, stationary phase also rise, because in total count we will count the living cells as well as dead cells, and in decline phase you will get a flat line because we will count dead cells also. And in case of viable count, flat, rise, flat and falls. In stationary case, flat means there is the newly formed cells are equal to the dead cells. Then special features of lag cells includes accumulation of enzymes and metabolites, and bacteria reach its maximum size. And in case of log phase, it is uniformly stained and metabolically active, and it is very small. And in case of stationary phase, it is gram variable, storage granules are producing, endotoxins, then antibiotics are produced, and in case of decline phase, then inversions are formed. The next is factors affecting growth of bacteria. So if you are inoculated a bacteria into suitable culture media, then bacteria needs some factors, then only the bacteria can grow. So these are the several environmental factors that affect the growth of bacteria, that include oxygen, carbon dioxide, temperature, pH, light and osmotic effect. First one is oxygen. Then some bacteria needs oxygen. Only in the presence of oxygen the bacteria can grow. Some bacteria will not grow in the presence of oxygen. Some bacteria can grow the presence of oxygen as well as absence of oxygen. Then coming to first one, that is obligate aerobe. Obligate aerobe means the bacteria will grow only in the presence of oxygen. So if we are keeping the bacteria in an anaerobic condition, then the bacteria can't grow. Example, Pseudomonas, Mycobacterium tuberculosis, Bacillus, Brucella and Nocardia. Second one is facultative anaerobes. Facultative anaerobes means they are aerobes, but they can grow in anaerobic condition also. Most of the pathogenic bacteria are coming to this category, that is facultative anaerobes. For example, E. coli, Staphylococcus aureus, Klebsiella, Streptococcus. These all are facultative anaerobes. That means it can grow in the presence of oxygen as well as absence of oxygen. Actually this is aerobes, but it can grow in anaerobic conditions also. Next one is facultative anaerobes. Facultative anaerobes means they are anaerobes. They can grow in the absence of oxygen, but they can grow aerobic conditions also. Example, Lactobacillus. The next one is microaerophilic bacteria. That means they can grow in the presence of low oxygen tension. Example, Camelobacter, Helicobacter and Mycobacterium bovis. Next one is obligate anaerobes. Obligate anaerobes means these bacteria can grow only in the absence of oxygen. Oxygens are lethal to the bacteria. This example is Clostridium. Clostridium can be Clostridium tetani, perfringens, botulinum, etc. That means these bacteria will grow only in the absence of oxygen. This bacteria can grow in anaerobic conditions only. If you are keeping in oxygen environment, then this bacteria will die. Then aerotolerant anaerobes, then it is tolerant to oxygen sometimes. Example, Clostridium histolytic. Next one is carbon dioxide. Then organism that require higher amount of carbon dioxide is called as carpinophilic bacteria. Example, Brucella abortus and Streptococcus pneumonia. Next one is temperature. So most of the bacteria will grow in the room temperature. So that is called as mesophilic bacteria, that will grow temperature within 25 degrees Celsius to 40 degrees Celsius. For example, most of the bacteria mesophilic, Staphylococcus, Streptococcus, E. coli, Salmonella, Shigella, then Vibrio, all these are mesophilic in nature. That means it will grow temperature between 25 to 40 degrees Celsius. If you keep the bacteria, suitable temperature for bacteria is 37, 38 degrees Celsius like that. Then some bacteria will grow in temperature below 20 degrees Celsius or in cold environment. This bacteria is called as psychrophils, and most of them are saprophytic in nature, example Pseudomonas. Then some bacteria will grow in high temperature region, that is 55 degrees Celsius to 80 degrees Celsius, example is Geobacillus stereothermophils. Then coming to the pH, most of the pathogenic bacteria will grow in pH 7.2 to 7.6. So the almost all bacterial culture media's pH will be like 7.4 to 7.5, 7.3 like that, because the ideal temperature, ideal pH for the growth of bacteria, most of the pathogenic bacteria is 7.2 to 7.6. These bacteria will grow only in this pH. That's why the culture media's pH is 7.4, 7.5 and 7.3. Then coming to the some bacteria, very few bacteria like lactobacillus can grow in temperature or pH below 5, and some bacteria will grow in alkaline pH, that is 8.2 to 8.9, example Vibrio cholerae. And next is light. Some bacteria can exhibit the phototropes, will grow in darkness, and these are sensitive to ultraviolet rays and other radiations in light. And photochromogenic mycobacteria produce pigments only in exposure to light. Then last one is osmotic effect. The bacteria are able to withstand wide range of external osmotic variation because of the mechanical strength of cell wall. We know that osmosis means there is a movement of solvent from higher concentrated area into lower concentrated area. That means sudden exposure to hypotonic saline may cause the cell shrinkage. If you are keeping a bacterial cell into high salt content solution, at that time 80% of bacteria is constitute of cell, that is cell constitute of water. So there is an disruption in the plasma membrane, so the water will come out from the cell, and the bacterial cell there is a shrinkage of bacterial cell, that is called as plasmolysis. Then we are exposed to distilled water may cause the swelling or rupturing of rupture, that is called as plasmoptitis. That means there is the water will come to the bacterial cell and bacterial cell become enlarged. Then coming to summary, then bacteria are unicellular prokaryotic microorganism. That means definite nucleus is absent, the genetic material is present in a diffuse manner, that is called as nucleoid, and it is the nucleus is absent and it is unicellular, but our human cells are multicellular and it is a microorganism. We can't see through our naked eye, which we can see through only microscope. And depending upon the shape, bacteria is classified into two shapes, cocci and bacilli. Cocci means round shaped bacteria, then bacilli means rod shaped bacteria. And bacteria is divided by a small process is called as binary fission. Binary fission means the first step is the chromosomes will replicate and it will move towards, then the bacterial cell will elongate and the mesosomes and plasma membrane will grow inwards, septa will form and it will divide into two daughter cells. And the time period between two successive cell division is called as generation time, and this generation time is different for different bacteria. For example, most of the pathogenic bacteria having the generation time is 20 minutes. For example, E. coli is 20 minutes. At the same time, Mycobacterial leprae generation time is 20 days. Then bacterial growth curve has 4 phases: lag phase, log phase, stationary phase and phase of decline. In lag phase, when you keep the bacteria into a suitable culture media, the bacteria will take up all the nutrients present in that culture media and it will reach its maximum size. There is no multiplication of bacteria in case of lag phase, so you will get a flat line, and in log phase there is an exponential growth of bacteria. So in lag phase, at the end of lag phase, bacteria reaches its maximum size. In log phase, the maximum sized bacteria will multiply into 2 daughter cells. Then in stationary phase, there is an equilibrium between newly formed cells and dying cells, so you will get a flat line. At the end, there is a depletion of the nutrients present in the culture media and accumulation of toxic substances, so all the bacteria present in that culture media will die, so you will get a phase of decline. Then factors affecting growth of bacteria including carbon dioxide, temperature, oxygen, pH, then osmotic effect, light, etc. Majority of the bacteria are mesophils, that means they can grow in room temperature, that is 25 to 40 degrees Celsius. Then most of the pathogenic bacteria are facultative anaerobes. Facultative anaerobes means this bacteria can grow the presence of oxygen as well as in the absence of oxygen. Actually it is anaerobes, but it can grow in anaerobic conditions also, and most of the pathogenic bacteria having pH 7.2 to 7.6, and some very less bacteria, few bacteria have grow in acidic temperature, that is lactobacillus, and some are growing in alkaline temperature, for example Vibrio cholerae, and bacteria except phototropes can grow in darkness. And these are the reference. Thank you.